Greetings to all of you. In this video lecture, I will be teaching you about diagnostic serum enzymes. The diagnostic serum enzymes are the enzymes which are markers of different conditions of the body, like cellular damage or different pathological conditions. So, the enzymes which are released into the blood serum by different organs of the body, by different cells and tissues of the body, are used by us for the diagnosis of different conditions or different defects or diseases of the body. We can define these diagnostic serum enzymes as they are the important enzymes that are used in diagnosis of certain disease or disorders of the body. Now, if we classify the diagnostic important enzymes, then there are two important types of diagnostic important enzymes. One of the enzyme is plasma specific and another category is plasma non-specific enzyme. Now let us see what are the difference between these two important diagnostic enzymes. These plasma specific enzymes are present in high concentration in the blood plasma. Okay, let me explain you in a different way. All right. Suppose this is liver and there are certain enzymes which are regularly released from the liver into the bloodstream. This is the bloodstream. So these enzymes are present in the blood and are used regularly for different specific purpose. So their concentration in the blood is in high. So they are present in the blood in high concentration. So required in the body in high concentration. Example is blood coagulation enzymes. So these enzymes are, this is one of the example of the plasma specific enzyme. They are the examples of plasma specific enzymes. So they are present in high concentration in the blood plasma. So if because of certain defects or disease their concentration becomes low in the blood, then it is an indication of the problem. Certain other enzymes are called as plasma non-specific enzymes. Plasma non-specific enzymes. Say for example, if I have shown you this heart or liver or some other organ, so there are certain enzymes which are present in the cells of these structures only, these organs only. So enzymes are found inside the body organs or body tissue. They are usually not released out. So if these organs are damaged in some disease, say for example kidney stone or heart attack, the tissues of these organs will be damaged. Then these damaged tissues and cells will release these enzymes into the serum. And so their concentration, which is these enzymes are usually present in low concentration in the serum, but now their concentration becomes high in the blood serum. So usually they are present in low concentration, but now when they are released from these organs or from these tissues into the blood, their concentration becomes high. So high concentration of non-specific enzymes is an indicative of certain problems of the body like heart attack or liver diseases, etc. In case of plasma specific enzymes, they are usually present in high concentration in the plasma because they are continuously or required release from the different organs of the body, like this in this example I have shown you with liver. So their high concentration is a normal case, but if their concentration decreases, they becomes low in the blood, then there is a problem. So this is the difference, main difference between plasma specific and plasma non-specific. Now let us move to our original sheet. As I told you about, there are two different types of plasma enzymes, plasma specific enzyme and plasma non-specific enzymes. When we say specific enzymes, they are the enzymes which are present in the blood plasma. So they are present in high concentration in the blood plasma. Now these enzymes, as I told you, have got specific roles in the blood plasma. So their decreased concentration is an indicative of the clinical importance. When plasma non-specific enzymes which are found inside the tissues of different organs of the body, when these enzymes are usually present in low concentration in the blood, they are present in low concentration in the bloodstream. But when any problem occurs like heart attack, their concentration increase in the bloodstream. The examples of non-specific enzymes are acid phosphatase, alkaline aminotransferase, creatine kinase, trypsin, SGPT and SGOT. We will be discussing these enzymes specially. The blood clotting enzyme, the ferredoxinase, pseudocholinesterase. Now these are the enzymes which are regarded as blood plasma specific enzymes, and they are present in high concentration in the blood plasma. Now let us go for the plasma non-specific enzymes when their concentration increases in the blood plasma, which is usually not there. So if we classify the diagnostically important enzymes, especially we are talking about these non-specific enzymes, then I have categorized these enzymes in 4 types. The diagnostic, first type is they are now these are regarded as markers. So certain diagnostically important enzymes or serum enzymes are markers of liver disease, biliary disease, bone disease, etc. Certain other are markers of skeletal muscles and pancreas defect, etc. Some other enzymes are markers of heart attack, that is cardiac markers. They are known as cardiac markers. And certain others are regarded as tumor markers and some specific other clinical importance enzymes. In this video, I will be taking you about only first 2 categories I will be teaching you for the sake of ease. The cardiac markers is dealt in a separate video, and the tumor markers and specific other enzymes are marked in another separate video. You can go ahead with that. In this video, we will be taking only the first 2 categories, A and B, the liver markers and the pancreas and skeletal markers. That's it. So now coming to the specifically with the enzymes in liver disease. So that means if a liver is showing some disease or damage and there are certain external symptoms in the patient, then these are the categories of the enzymes which are found during a liver disease like liver cancer or gallstones or anything else. First enzyme is ALT, that is SGPT. The second enzyme is AST, that is SGOT. Then we have got alkaline phosphatase, 5 nucleoside transferase, and gamma glutamyl transferase, that is GGT. The last 2 enzymes in this category are actually biliary tract enzymes. That means they are released from the cells of the bile duct. But first 3 enzymes, these enzymes are released from the liver and some other tissues also, the cells of the liver, not by the duct. Now let us discuss all these enzymes one by one in a quick succession. ALT, the full form is alkaline transferase. Now this is also known as SGPT because the another name is serum glutamate pyruvate transaminase. The enzyme commission number is 2612. Now what is the role of this enzyme in the body? This enzyme combine alanine with alpha ketoglutarate and convert these two into pyruvate and glutamate. So this is the physiological role of this enzyme ALT and SGPT. It works with a coenzyme called as paradoxal phosphatase. It is a coenzyme which works with ALT, that is alkaline transferase. Now normal range of alkaline transferase in the blood is 5 to 35 unit per liter or international unit per liter. Now, during liver problem, there is high concentration of this enzyme in the blood serum. So this enzyme is found in liver in high concentration, but when the liver disease occurs, the liver release this into the bloodstream, and it is also present in heart and skeletal muscles, but the role is not very specific. So specifically in liver disease it is been detected. Now what happens with this enzyme? The liver, if it is damaged because of cirrhosis or cancer or hepatitis or any other infection, if the liver cells are damaged, then they start releasing SGPT, that is this enzyme alkaline transferase into the bloodstream, and then their concentration in the bloodstream increases and it becomes a marker for us. We go for the blood test. If the concentration of SGPT is high in the blood, it is an indicative of the liver problem. Then the second enzyme which act as a liver disease marker is AST, aspartate transferase. This is more a cardiac marker than a liver marker, but it is also detected in a liver. That's why we have to explain it. It is also called as SGOT, serum glutamate oxalate transferase. This is another name. The role enzyme commission number is 2611, and the role is it combines aspartate with alpha ketoglutarate, and this enzyme converted into oxalate and glutamate. This is the physiological role of this enzyme in our body. Now, this enzyme is usually found in the cytosol and mitochondria of cardiac cells, cardiac muscles, and also in the liver, especially in the heart muscles, and also liver also it is present. Its normal range in the blood serum is 8 to 20 units per liter. Now, what happens in this case? Uh, if the concentration of SGOT increases, it is more an indicative of myocardial infarction followed by muscular dystrophy, and then it is also an indication of the liver disease. So, we say the AST is more specific for heart disease than liver. Alright. So, if we talk of these first two enzymes, that is ALT and AST, these two enzymes which we have discussed just now, these two enzymes level increases in heart and liver, both the diseases. But ALT, the first one, is more specific for liver disease than heart, but the second one, that is AST, is more specific for cardiac disease than the first one. Alright. So, coming on to the third enzyme, that is alkaline phosphatase. In short, it is written as ALP. Now, this alkaline phosphatase is actually a group of enzymes that hydrolyze phosphate at alkaline pH. Whenever the pH is alkaline, the substances will be separated from their phosphate. So, it hydrolyzes the phosphate from different substances. It is found not only in the liver and heart, but it is also found in intestinal epithelium, kidney tubules, that is the tubules of the kidney, bone, liver, and placenta. Since it is present in liver also, it is a marker of liver disease. Now, increase in the concentration of ALP in the bloodstream mainly occurs in liver and bone disease, especially they are present. So, if we see this chart, hepatobiliary disease are the different liver diseases, that is liver stone or liver cancer or anything, any problem with the bile duct, it is hepatobiliary disease. ALP increases 2 to 3 times in these disorders. In case of bone disorders, bone cancer, osteomalacia, rickets or Pigott's disease, their concentration increases 10 to 20 times. So, ALP increase is very much high in case of bone diseases. This Pigott's disease is a disruption of replacement of old bone tissue by the new tissue. All the time the new tissues are being formed new bone tissues are formed, and the old bone tissues are replaced with the new tissue. And if any obstacle occurs in this case, then this is called as Pigott's disease. Okay, all right. Then we have got biliary tract enzymes. There are certain enzymes which are released not by the cells of the liver but by the bile duct, that is biliary tract enzymes. There are two important enzymes in this category. One of the enzyme is 5-nucleotidase. It is also called as nucleotide phosphate or NTP, and the second one is gamma glutamyl transferase, GGT. It is also written as. Then first one is 5-nucleotidase. Now, the role of this enzyme in our body is converts AMP with this enzyme, that is NTP is the short form. AMP is broken down into adenosine and its phosphate is separated. This is a physiological role. Now, this enzyme is found in the cell membrane of bile duct, especially cell membrane. The normal range in the blood serum is 2 to 18 unit per liter. If it is increases, then there is a problem. The concentration of this enzyme NTP increases in cholestasis. This is a problem when bile flow in the bile duct is imparted because of the gallstone or cancer. So, because of gallstone or cancer, there is blockage in the bile duct, and so the bile is not able to come out of the liver, and then it is not able to flow into the duodenum, and this is called as cholestasis. So, during cholestasis, again this concentration of nucleosidase increases, and so it becomes a marker. Now, another enzyme which is released from the cells of the bile duct is gamma glutamyl transferase. This is another enzyme. Now, this enzyme is present in the cell membrane of these ducts, and what is the role? it transfers gamma glutamyl group from proteins to other substances, another acceptors. So if there is a protein, it will transfer the gamma glutamyl group. I am not going to show you the structure for that. Gamma glutamyl group, it transfers from one protein to the other acceptors, and it also helps in the synthesis of a substance glutathione. All right. So the normal range in male and female, it is different, 2 to 30 units per liter, and in female it is 1 to 25 units per liter. These are the differences in the normal range. But beyond this normal range, if it is increases, then it is an indicative of the liver stone or bile duct problems. Now there are certain enzymes. Now this is all about the uh, markers of liver disease. Now coming into the next category, that is the pancreas. If pancreas show some defect, then there are certain enzymes which becomes higher in the serum also. The serum enzymes of pancreas. Such enzymes are used to diagnose pancreatic disease. Say for example, alpha amylase, lipase, and trypsin. They are the common enzymes released by pancreas, and if any problem occurs in the perilium, then there is a defect in the normal concentration of these enzymes. Now, if you talk about the first enzyme, that is alpha amylase, it is secreted by pancreas. Now, what happens? This enzyme is actually degrading the polysaccharides. Uh, uh, More correctly, it breaks down the alpha 1,4 glycosidic bond, but not 1,6. If you have seen the structure of polysaccharide, glucose, 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 glucose. So this is 1,4 glycosidic linkage. Now this 1,4 glycosidic linkage is actually hydrolyzed by this enzyme. But if side group of, inside the glucose molecules are present in the polysaccharide, then this linkage is actually 1,6 linkage, and it is not been affected by this enzyme. So this enzyme only affects the 1,4 linear linkage, but not the 1,6 side linkage. Now it is also present in the urine because this enzyme, if present in the blood, can pass through the glomeruli of the kidneys. Normal range is 50 to 120 unit per liter. Alpha amylase is a marker of pancreatitis. It is a tumor of pancreas. That means the pancreas, when becomes tumorous, then their concentration increases. Another enzyme secreted by pancreas is the lipase. It converts triglycerides into monoglyceride and fatty acid, and the normal serum level is 2 to 10 unit per liter. Its concentration increases in these conditions, pancreatitis also, and cancer. The last enzyme which is here is trypsin. It is released by pancreas. It hydrolyze. This enzyme hydrolyze the carboxyl group of lysine and arginine with other amino acids. That is a physiological role. Now, presence of trypsin in serum is a defect of cystic fibrosis in the first 6 weeks of the life, and then later on it becomes normal. Now, this is all about the different enzymes which are used uh, as a marker for pancreatic disease. So, let me recollect what